Hello, everyone. In the next lecture in this lecture series on transcriptomics, we will discuss how to determine primer efficiency for qPCR. So I will briefly start again with an overview of the basic principles of qPCR. So as a classical PCR cycle, a qPCR cycle consists of three phases, which are, first of all, denaturation, subsequently primer annealing, and finally primer elongation. The difference between classical PCR and real-time PCR is that during real-time PCR, as the name implies, the generation of the PCR product is monitored in real time. And this is done through the use of a DNA binding fluorescent probe. And the most important ones that are frequently used are Cyber Green, Tuckman, and molecular beacons and scorpion probes. So as I mentioned previously in this lecture series, we specifically focus on qPCR using Cyber Green. So I will briefly explain the principle of Cyber Green again. So what this molecule will do is it will intercalate between two strands of double-stranded DNA. And when it does so, it will strongly increase its fluorescence. So you can see this here. When a Cyber Green is freely available in the solution, it emits very low fluorescence, whereas upon intercalation between double-stranded DNA, its fluorescence intensity will very strongly increase. And it's this fluorescence intensity that's monitored in real time throughout the different qPCR cycles. And Ava Green is a very similar dye compared to Cyber Green, similar working mechanism, but it has an improved performance. As I previously mentioned, the use of these probes has several advantages. So first of all, it's relatively simple. They are less expensive compared to certain other probes like Tuckman. And the nice thing is that they can also be used to quantify the expression of any gene. The disadvantage on the other side is that they are non-specific, which means that they will bind to any double-stranded DNA, so also to primer dimers and other aspecific products. And in case this occurs, the obtained results are unreliable. But as you might remember from the previous lectures, this is something we can test using a dissociation curve. Then the main principle of qPCR is that a CQ value will be determined for each sample, and this CQ value will allow us to compare gene expression levels between different samples. The CQ value is also often called CT value. These stand for quantification cycle or threshold cycle. And these are the number of cycles that are required for the fluorescence in a certain sample to reach a predefined threshold. This threshold is shown as a horizontal orange line here, and it's generally placed in the middle of the exponential phase of the reaction, which is also called the log linear phase. And the CQ value is inversely proportional to the gene expression level in a sample. So if the expression of a certain gene in a sample is high, the CQ value will be low. On the other hand, if the expression of the gene is relatively low, the CQ value will be higher. The reason for this is that when the expression of a gene is low, there are only a small number of copies of the messenger RNA, so also only small number of copies of the cDNA, which means that more PCR cycles are required for the amplification of the product in order to reach a certain amount of PCR product, which corresponds to a certain amount of fluorescence, so in this case, the threshold level of fluorescence. So these are, again, the main principles of qPCR. And as we saw in the previous lecture, we need to design primers for the specific genes of which we want to measure the expression during qPCR reaction. And for designing these primers, there are different parameters that we need to take into consideration. And these were the amplicon length, the primer melting temperature, the self-complementarity of the primers, and also their binding location. And we discussed in a previous lecture how to take into account these different parameters. I also told you that you can then design primers using different online tools, for example. But even though primers might meet all of these requirements, as we discussed during the previous lecture, this does not necessarily mean that these primers will also perform well in the lab, in real life. So even though on paper these might be perfect primers, it's still possible that in the lab they do not perform well. And therefore, it's very important to test the efficiency of the primers. So you will always design primers in a pair. So you have one forward primer and one reverse primer, which will together lead to the amplification of a specific PCR product. And what does primer efficiency now mean? Well, if this primer pair has an efficiency of 100%, this would mean that the amount of PCR product, so the amount of amplicon, will double with each PCR cycle. In case this primer efficiency would be too high, or on the other hand, too low, this would lead to inaccurate quantification of gene expression levels. Because, as you will see in the next session on data analysis, the method that we will use to compare gene expression between samples is known as the 2 to the power of minus delta CQ method. And this 2 here, in this formula, actually refers to the doubling of the product with each PCR cycle. So the amount of product is multiplied by 2 in each cycle, hence this 2 here in the formula. So this formula assumes that the primer efficiency is 100%. Of course, small deviations from this value of 100% would not be a problem, but you can imagine if in real life the primer efficiency is only 50% or maybe 200%, then this method used for data analysis would not give us accurate results. So therefore, it's very important prior to using primers to really measure the expression of a gene of interest in a certain set of samples to first test the efficiency of these primers. Now, how do we do this? This is the general procedure that is used. So what you will do is when you have a certain batch of samples, so cDNA samples, what you will do is you will generate a pooled sample of these cDNA samples. So pooled sample means that from each of your cDNA samples, you will add a certain volume. So let's say you will have 20 cDNA samples, then you might, for example, take 1 microliter of each cDNA sample and add those volumes together in a new tube. Then from this pooled sample, you will prepare a twofold dilution series. And this is what you can see here. So here we will start with our pooled sample. So this would be the undiluted sample. And what you would do then is in a serial dilution series, you would first dilute the sample twice. So you would have a 1:2 dilution. You would dilute that one again twice, so you would obtain a 1:4 dilution, then a 1:8, 1:16, 1:32, and 1:64. So in this way, you obtain this serial dilution series. And subsequently, you will use this dilution series for primer efficiency testing. And you do this simply by performing a qPCR reaction with your primers of interest on this dilution series. So instead of just using your 20 individual cDNA samples to perform the qPCR, you instead perform the qPCR reaction on this dilution series. Then the result that you will obtain will look like this, hopefully. So what you again see here are amplification curves, in this case for the different dilutions in the series. On the x-axis, you can see the number of cycles that have passed during the qPCR reaction. And on the y-axis, you can see the cyber green fluorescence plotted on a logarithmic scale. What you would expect now is that in case the primer efficiency is 100%, that there would be a difference of 1 CQ value between each subsequent dilution. Because if you start from a 1 on 2, so a twofold dilution, in comparison to a non-diluted sample, you would only have half the amount of starting material. The amount of cDNA for that specific gene that you're interested in would be half the amount in the undiluted sample. So in a twofold diluted sample, it's half the amount that you find in the undiluted sample. And hence you would need an additional PCR cycle to reach the same amount of DNA, so the same amount of fluorescence. And one additional PCR cycle would mean that here you have a shift of 1 CQ value. So here I explained this in this table with an example. So here you start with the undiluted pooled sample. Then you have the 1 and 2 diluted sample, 1 and 4, 1 and 8, 1 over 16, 1 over 32, and 1 over 64. So the amount of starting material for the qPCR reaction will be divided by 2 with each step. So for the 1 and 2, it's half of the undiluted sample. For the 1 and 4, it's again half of the 1 and 2 diluted sample, and so on. And then let's say you would have a CQ value of 15 for the undiluted sample, then you expect, in case primer efficiency is 100%, that you will need 1 additional cycle to reach that same threshold for a sample that only contains half the amount of starting material. So this means 1 additional CQ. So in principle, with each step in the dilution series, you would expect the CQ value to shift to a higher value, which is 1 higher than the previous CQ value. And this is something that you can first of all visually inspect. So when you obtain your amplification curves, you can have a look at them. And in this case, it seems okay because you see that the different curves are nicely separated by approximately 1 CQ value. So if the distance between the curves would be much larger or much smaller, that would imply that the primer efficiency strongly deviates from 100%. So how will you then calculate the primer efficiency? Because you can, of course, check whether the amplification curves visually look okay, but what we want is ultimately to be able to put a certain value on the primer efficiency. So therefore, what we will do is we will plot the obtained CQ values against the logarithmic or the logarithm of or dilutions. So here you can see this graph. So here on the x-axis, you have the logarithm of the dilution. And on the y-axis, you can see the CQ value. Then from these data in Excel, for example, you can generate a so-called standard curve. So you will draw a trend line through these data points. And then from this trend line, you will calculate the slope. And this slope you will then see in the formula of the trend line. Then next, you will calculate the primer efficiency using the following formula. So the formula is 10 to the power of minus 1 divided by the slope. From this value, you will subtract 1, and then you will multiply the value by 100. Then you will hopefully obtain a value that's close to 100%. So in general, we say that primers with an efficiency between 90 and 110% are considered suitable for qPCR. But some people are also less strict and they allow deviations that are a bit larger. So they indicate that primer efficiencies between 80 and 120% are okay to use. But But what is also important is that you should also check the R square, so the correlation coefficient of the standard curve that you generate. And this should be above 0.99. So this value indicates how well the trend line fits through the different data points. Because what might be possible is that based on the slope of the trend line that you generate, the primer efficiency might be okay. It might be, for example, 105%. But if all the individual data points very strongly deviate from this trend line, that would most likely indicate that there is an issue with the dilution series. Maybe you didn't pipe it very accurately. And in that case, yeah, this trend line and also the slope that you obtain from it is not very reliable. And yeah, it might be difficult to say whether the primer efficiency that you calculated is really the correct one. So therefore, it's always important to also check the R square value. So now you know how to calculate primer efficiency. Later on, there will also be a demo of how to calculate this with some real-life examples. But what I want to discuss next is which parameters can affect primer efficiency. And here I listed the 4 most important ones. These are the annealing temperature, the primer concentration, the magnesium concentration, and the presence of PCR inhibitors in the cDNA samples. And we will now discuss each of these in the next slides. So starting with the annealing temperature. So as I mentioned in the lecture on primer design, the annealing temperature is the temperature that is used during the annealing step of the qPCR program. So the temperature at which, or the temperature which you will use to let the primers bind to their template. And usually this is 60 degrees, or at least in the range of 60 degrees. So this is close to the melting temperature of the primers, which we discussed in the previous lecture. What would now happen if the annealing temperature is too high? So in that case, the annealing temperature would be much higher than the melting temperature of the primers, which means that at this temperature, the primers would already denature from their template. So they would not be able to bind to their template, and hence no PCR product will be formed. On the other hand, when the annealing temperature is too low, primers will also be able to bind to other regions in the cDNA besides their target gene. This is because when the annealing temperature is too low, this will also allow for some mismatches. So the primer and the part that it will bind to will not be fully complementary to each other. And this will then lead to the formation of non-specific products, which is also something you want to avoid, because in that case, the CQ values you obtain are non-reliable. Now, in principle, you could adjust the annealing temperature to optimize the primer efficiency. And this is something you could, for example, do using a classical PCR. So in a regular PCR device, not a qPCR device. And what you could do in such device is to perform a so-called gradient PCR. Gradient PCR means that in each column of the device, or in each row of the device, you will perform exactly the same PCR run. The only thing that will differ is the annealing temperature. And then you will put the resulting product on a gel. So you will perform gel electrophoresis, and then what you will obtain is something like this. So you will see that at lower annealing temperatures, you might get the formation of a-specific products, as we mentioned. So you will see that there is multiple bands on a gel. On the other hand, at an annealing temperature that is too high, you would not obtain any product anymore. So there you would not see a band on the gel. So you're specifically looking for a temperature where you only see one band in a gel, such as, for example, here at 60 degrees. However, Using different annealing temperatures for different primer pairs, so for different genes of interest, is not ideal, because this would mean that you would have to measure each gene of interest on a separate qPCR plate. Of course, when you have a large number of samples, this might not be a problem, but if you have a relatively low number of samples, what's very interesting is if you could measure the expression of different genes on the same qPCR plate, because this could save you a lot of time. So therefore, you should ideally have primers that all have a melting temperature close to 60 degrees, because this would allow you to measure the expression of different genes on the same qPCR plate. Another parameter that affects primer efficiency is the primer concentration. So typically qPCR reactions use primer concentrations between 0.1 and 1 micromolar. What will happen when a primer concentration is too high? Well, this might increase the risk of primer-dimer formation, because the more primers are available, the higher the likelihood that they will also start binding to each other. On the other hand, when the primer concentration is too low, there might be insufficient primers to amplify all the available cDNA templates, and hence you would reach the plateau phase earlier, which is also something you want to avoid. So in case primer efficiency is not optimal, so it's not in the range of 90 to 110%, you can try to increase or decrease the primer concentration and test which effect this has on primer efficiency. The next factor that influences primer efficiency or PCR efficiency is the magnesium concentration. And in qPCR reactions, typically a magnesium concentration between 1 and 4 millimolars is used. Now, what is the function of magnesium in qPCR? Well, first of all, it serves as a cofactor for the DNA polymerase. So this is something you can see here. Here you can see the active site of the DNA polymerase, and you see that there is magnesium ions present here. And in addition to that, magnesium is also known to stabilize double-stranded DNA. Now, what will happen if the magnesium concentration in the reaction is too high? Well, this might cause formation of non-specific products because double-stranded DNA will be stabilized, even in case when there are some mismatches. Or on the other hand, if the magnesium concentration is very high, this might even interfere with DNA denaturation. On the other hand, when magnesium concentration is too low, the DNA polymerase will not be sufficiently active because it requires magnesium as a cofactor to properly work. And in that case, no PCR or too little PCR product would be formed. However, what is important to keep in mind is that magnesium concentration is usually difficult to adjust because, for example, when you purchase Cyber-Green from a company, this Cyber-Green is not just the fluorescent probe, but it also the solution also includes other factors such as magnesium, but also the nucleotides that are required to amplify the product. It also contains the polymerase, so it's already a mixture of different components. So the magnesium is already part of the Cyber-Green solution, so it's not something you can really adjust. And then the last factor that will influence PCR efficiency is the presence of PCR inhibitors in the cDNA samples. So PCR inhibitors will inhibit the DNA polymerase and or the annealing of primers to the template. Examples of PCR inhibitors are certain proteins, but also products that are used during the RNA extraction, such as detergents, salts, ethanol, or also polysaccharides that could be present in the samples. So therefore, it's very important that after the RNA extraction, you always test the purity of the RNA. Remember that you can do this, for example, by using the nanodrop and determining the 260/280 and 260/230 ratios, as too low values there will indicate potential presence of PCR inhibiting compounds. Now, what is maybe a bit counterintuitive is that when there is a large amount of PCR inhibitors present in your cDNA samples, this will artificially increase the primer efficiency above 100%. So this doesn't really mean that your reaction becomes more efficient, but it's just a consequence of the way in which we determine primer efficiency. So remember that we determine primer efficiency on a dilution series of a pooled sample. Now what will happen is, let's say for the undiluted pooled sample, you would expect a CQ value of 15 in case no inhibitors were present. What will happen in the presence of PCR inhibitors is that the CQ value will become higher because the inhibitors inhibit the PCR reaction, and hence you will require more PCR cycles to reach the same amount of DNA and hence the same amount of fluorescence. So this means there is a shift in CQ value, and for example here the CQ value would increase by 1 from 15 to 16. For the twofold diluted sample, there will still be a shift of the CQ value to a higher level, but the increase will be smaller in this case because when you dilute this pooled sample, you do not only dilute the cDNA, but you will also dilute the PCR inhibitors that are present in the sample. So the total amount of PCR inhibitors will be lower in this twofold diluted sample compared to the undiluted sample. Hence, the shift in CQ value between a sample without inhibitors and with inhibitors will be smaller. So here it might shift from 16 to 16.7. With the next dilution step, you are again diluting also the PCR inhibitors that are present in the sample. And hence the shift in CQ value will again become a bit smaller. So here you will have a shift from 17 to 17.5. And this happens with each subsequent dilution. So what you will see is that the effect of the qPCR inhibitors on the obtained CQ value will become less pronounced in more strongly diluted samples because the PCR inhibitors are being diluted as well. And at some point, for example, here for the 1 in 32 dilution, the inhibitors will not have any effect anymore because they're also so strongly diluted at this point that they don't really interfere with the PCR reaction anymore. So here the CQ value will remain unchanged. But what you will see then here is if you would have these samples without PCR inhibitors, you nicely have a CQ value difference of 1 between each subsequent dilution. But in this case here, you see that here, especially at the beginning, the difference in CQ value is lower than 1. So here there is only an increase of 0.7. Here the increase is 0.8 and so on. So here the differences in CQ value between subsequent dilutions are smaller than 1. And as a consequence of that, you will have a PCR or primer efficiency, which is higher than 100%. But this is just a consequence of the method we use to determine qPCR efficiency. So this was everything I wanted to tell you regarding calculation of primer efficiency. So what I will do next is I will give a demonstration of how to calculate primer efficiency using an example, and then you can also calculate primer efficiencies in some exercises.